Hello students, welcome to EPG Pathshala. I am Rohini Muthuswamy from the School of Life Sciences, JNU. Today we are going to discuss about a module, protein-protein interaction and its requirements, protein microarray, genetic engineering, under the paper, genetic engineering and recombinant DNA technology. The learning outcomes of this module are, we're going to understand what is protein-protein interaction, what type of protein-protein interactions are present, factors regulating protein-protein interaction, structural characteristics of interacting protein partners, the specificity of protein-protein interactions, conformational change on protein-protein interaction formation, multi-specific protein-protein interactions, evolution of protein-protein interactions, diseases caused by disruption of protein-protein interactions, and techniques to study protein-protein interactions. So the protein-protein interactions, you will find that all processes in the cell are mediated by protein complexes. Very rarely does a protein act on its own. It will form some kind of a complex with other proteins, and these complexes are not held together by any covalent bonds, but they are held together by non-covalent interactions, which really makes this, the complexes can come together, they can fall apart. This is a dynamic process that keeps on happening within the cell, and any interaction which is disrupted, if the interactions become abnormal, that will result in diseased state. For example, in case of Huntington's disease, you have protein-protein aggregation formation that results ultimately in the disease manifestation. The protein-protein interactions can be classified into, there are many different classifications we can use, but primarily we can classify them into a homodimer or into heterodimers. Many restriction enzymes, for example, they will form as a homodimer. Alkaline phosphatase forms a homodimer. But then there can be also interactions which are heterodimer in nature, which is to say two different proteins come together to form a complex. For example, the c-MYC forms a complex with MAX protein, and these two together act as a transcription factor regulating the gene transcription of many downstream effector genes. The interactions can be also classified as either something called as a stable obligate dimer, in which case it means that the protein cannot exist by itself as a monomer in solution in the cell. It will always form a dimer. This could be because the protein is inherently stable and the stabilization happens only if it is forming a dimer. Most of the complexes in the cell are transient. They are non-obligate dimers. They come together, there is some kind of a force that holds them together, they perform their function, and then they can fall apart. In fact, if you look at transient protein-protein interactions, you can have weak transient interactions where the KD is around micromolar range, or you can have a very strong transient and it is the high affinity more towards a nanomolar. But a nanomolar affinity is a permanent interaction. That is where you have an obligate stable dimer formation happening. So what are the factors that regulate protein-protein interactions? There are three major factors that regulate these interactions. One is a concentration of the interacting partners. The second is a physicochemical environment, and the third is the encounter. So the protein-protein interactions are very important because this is how the cellular processes work. They are not carried out by single proteins. There are these complexes that come into play. These complexes can be either very stable or they can be, transient. They can come apart and then come together. So this complex formation is what drives a cell, and this is what we are going to study in this module is what are the parameters of the protein that is required for forming these complexes, and how do we study these complexes? How do we figure out that there is a protein complex that has formed? So this is what is going to be the focus of this module. So of the factors regulating the protein-protein interaction, the concentration of the interacting protein is very critical. This is because the proteins, once it is formed in the cell, they are not always going to be there. The concentration will vary. there are many factors it is regulating. First of all, the gene expression itself. If suppose there is a signal that comes in and that says, look, this protein needs to be produced in the cell, then that particular gene transcription will be turned on. You will have a high amount of protein concentration in the cell. The second level of regulation of the concentration could be how the mRNA is. Is the mRNA stable, or is it a rapidly turning over mRNA, in which case the concentration of protein can fluctuate. It can be also that the mRNA is being targeted by an RNA interference process, so the protein production is being reduced. Then the protein itself can be activated by proteolysis, like for example caspases. Remember, caspases are produced as procaspase and then it is activated when the signal comes that the cell has to die. Then the procaspase converts itself into caspase by a proteolytic activation. Then it can be the binding and subsequent folding can control the concentration of the protein. Translocations for the optimal activity. So a protein might have to be translocated to the plasma membrane, then only it will be active. So the concentration of the protein on the plasma membrane will depend upon the transport process. Then the protein itself can undergo rapid turnover because it can be ubiquitinated and then proteolytically cleaved. So the concentration of the proteins in the cell is not ever dynamic. The concentration of the interacting proteins can keep on changing within the cell. So the physicochemical environment, why they are important? They are important because interactions can be stabilized by ions. For example, calcium is required for calcium binding proteins. Magnesium is required for many DNA proteins that mediate DNA processes like DNA polymerase. So, and if this a particular ion is not present, then the protein folding will be altered and therefore the protein-protein interaction will change. So therefore the interactions are stabilized by these ions. Sometimes interactions are stabilized by small molecules like for example ATP or biotin, and the availability of these factors therefore affect the protein-protein interactions. The third parameter that you have to worry about is the encounter. Remember, this is a cell, so you have proteins going about and they are diffusing, they are moving, they are randomly moving, and the two interacting proteins have to actually meet with each other, they have to collide, then only the interaction can happen. And the factors that influence this encounter are relative diffusion, which how they are moving about in the cell, what is their diffusion rate, then the concentration, of course, and then finally, are they present in the same place or are they present in different this? So the localization is another factor that influences the encounter. So now the two proteins have interacted with each other. What is the structural characteristic of these interacting proteins? The protein-protein interactions are defined by four major structural parameters: the size of the contact area, the shape, the number of amino acids, and then of course the polarity of the interface and segmentation pattern. And we will now go ahead and look at each of these. So the size and the shape of the contact area is a critical determinant in the interaction proteins. So for example, if you look at the interaction between protein A and protein B, studies have shown that the interface between the contact area is almost always larger than 1100 angstrom squares. And each protein contributes about equal, so about 500 to 600 angstroms is what is contributed by each of the protein. The other thing that the studies have shown is in an obligate permanent interaction, the interfaces are much larger than in non-obligate complexes. And further, the contact area tends to be very flat. So these are some of the structural characteristics of the interacting contact area. The other thing that people have done is to look at amino acids are contributing to the contact area. So for the 1100 angstroms, generally about even 2 amino acids will be sufficient. But the studies have shown that at least 34 plus minus 7 amino acids are involved in the interaction. And the interactions are primarily mediated by hydrophobic interactions, so the water molecule tends to be excluded. There is very less of electrostatic salt bridge kind of formation, and the amino acids in contact with each other, they do not form a contiguous segment. So it is not that, you know, this entire region of say amino acids 1 to 7 are going to be, or 1 to 34 are going to be interacting. So 1 to 2 can come, 1 to 4 can come from one area, then the 5, 6, 7 can come from another area, and so it is not going to form a contiguous segment, but it is going to be discontiguous. So the other parameter you have to worry about the specificity of the interaction. You have in the cell, say A, B, C, D. What determines only A and B will interact to form a complex? So this specificity of interaction is determined by the shape of the contact area, the thermodynamics of the interaction, and the third point, the localization. So the thermodynamics of the interaction, of course, we all know that Gibbs free energy delta H, G equals to delta H minus T delta S, and interactions are favored when delta G is less than 0. This is a given fact. But the delta G, there are two parameters that we have to consider. One is the delta H and the other is the delta S. The thing is that when we talk about interactions, we already know it is favored by hydrophobic interactions. The water is excluded out and the salt bridges are very rare. So the enthalpic changes are contributed by the hydrogen bonding, electrostatic interactions, and van der Waals interactions whenever and wherever happens. But for a protein-protein interaction, the major contribution is going to be your entropic changes. There are two things that will contribute to your entropic changes. One is the loss of the free motion as proteins come closer together. So if you have protein A and protein B in cell, they both will be tumbling, and there are a lot of motions that is possible if they are existing by themselves. But when they start coming closer to each other and forming an interacting molecule, the number of motions that is available to them, the possibilities, the number of vibrational motions as well as the rotational motions that is available to them, that decreases, and this contributes to your entropic change. And then when the proteins come together, the water molecule is excluded out, so your interaction is favored by these hydrophobic interactions. So these hydrophobic interactions are also going to contribute to the entropic changes. In fact, the hydrophobic interactions are completely dependent upon the entropic changes. So therefore, this entropic change becomes the bigger contributor in certain ways to the delta G. It is not to say that the enthalpic changes do not contribute, they also contribute, and together these two is going to make sure that the delta G is less than 0. And only when that happens will you have the interaction, a stable interaction, if you will, in whatever sense we talk about, whether it is just for that moment it is going to come together or it is going to be always together, whether we talk about stable obligate complexes or we talk about non-obligate complexes. The localization of the proteins also play an important role in protein-protein interactions. If suppose two proteins are not in the same place, obviously they cannot interact, right? So let's take an example of NF-kappa B. Remember, NF-kappa B can exist both in the cytoplasm as well as in the nucleus. In the cytoplasm, it is held in an inactive form because it forms a complex with I-kappa B. When the signal comes, I-kappa B is degraded and then only B can move into the nucleus to mediate its action. So the localization of the protein where it is present determines what kind of a complex will it be forming. The other parameter that we have to worry about is the conformational change on protein-protein interactions. So it depends upon the protein-protein interactions. In some interactions, there might not be any conformational changes, but in other interactions, there can be conformational changes. In some, only the side chain movement has been observed, whereas in others, the entire segment movement has been observed. So it cannot be determined a priori. We cannot just look at two proteins and say this is how they're going to be interacting and this is what is going to happen. But this is an experimentally you have to determine what happens to the conformation of the protein or both the proteins on interaction. The other major important factor is that many proteins can form multi-specific interactions. So for example, in this example, we are showing beta-catenin can interact with many different proteins. There are of course proteins that form only very specific interactions, and they might be interacting with only one other protein. But things like beta-catenin, pCNA, which is involved in DNA replication, they interact with many different proteins, they form many different subunits. Many transcriptional co-activators and co-repressors also form this kind of an interaction. The multi-specific interactions are governed by structural properties of the interfaces and the thermodynamics of the interaction. Many different factors come into play to determine whether a protein is going to form multi-specific interactions or it is going to be monospecific interaction. Finally, why would protein-protein interactions happen? If you have one single protein that can carry out its function, why did the systems evolve protein-protein interactions? There are many different hypotheses that have been put forward. The two major hypotheses is that one is that it increases the stability of the protein. This is especially true when we talk about stable obligate complexes, because there the protein monomer cannot exist. It is unstable by itself. So it has to form a dimer. The other hypothesis that has been put forward is it optimizes the efficiency of their function. So if suppose you have a multi-subunit complex, protein complex, and you have many different proteins there, and each protein might be doing one specific function. A classical example is the general transcription factors, where you have TF2B, TF2D, A, F, all these things come into this, and each protein has a specific function that it does. So to optimize the efficiency of the function, there could have been the evolution of protein-protein interactions. So protein-protein interactions are very important for cellular processes, and disruption in the protein-protein interactions have been linked to diseases. Both the disruption as well as formation of abnormal interactions. For example, in Alzheimer's disease, we have the amyloid plaques formation, or in Huntington's, we have the Huntington proteins aggregating together. So here there is this abnormal interaction that is happening because there are certain patches in the protein that is allowing this protein-protein interactions to take place. And this causes large aggregations, and these aggregations can either accumulate in the cell and create problems, or they can sequester other proteins and then cause a loss in function. So there is this protein called alpha-1 antitrypsin, which is present in our systems. Persons, non-smoking people, what happens is the alpha-1 antitrypsin keeps the alveolar in a normal state. But in the smoking individuals, the alpha-1 antitrypsin gets oxidized because of the increased production of ROS, and this then causes an imbalance in the system, leading to the breakdown of the alveolars in the lung and causing the pulmonary COPD kind of a state. So the disruption of the protein-protein interactions as well as the aggregation of protein-protein interactions can lead to diseased state. So protein A and B interacts. How do we know A and B interact? How can we detect it? Or the other parameter that we have to think of, you have protein A. How do we know what are the interacting partners of A? Does A interact with B, C, D, E, or does it interact with C, D, E, F? So how can you identify all the proteins that this particular protein is interacting with? So these are two different parameters. So one involves global genome-wide methods that helps you identify all the protein partners, and the second is to look at the individual protein interactions between A and B, between C and D. So that is what is the individual protein interactions. So the genome-wide protein-protein interactions enables you to understand the entire proteome. I mean, how does what are the other proteins that it interacts with? So there are two major methods used for this process. One is a protein microarray, and the other is the affinity chromatography coupled with mass spectrometry. So there are two types of protein microarrays that have been described. One is an analytical microarray, and this cannot be actually used for protein-protein interactions. Here the antibody is immobilized, and you basically try to identify the kind of proteins that is being expressed in a cell. But the functional microarray is the one that is used extensively for protein-protein interactions. Here the protein is immobilized, and then you ask what kind of proteins present in the cell lysate are able to interact with the immobilized protein. So in case of protein arrays, we have the antibody immobilized, and we are looking for what kind of antigen is interacting or protein is interacting with this antibody. And it is basically, this is used to check for protein expression level, protein profiling, diagnostics. The protein microarray, which is a functional microarray, here what we do is we are basically immobilizing the protein, and then we are asking, using protein probes, what kind of proteins are interacting with it. This can be actually many different things can be done. You can use protein probes to do protein-protein interactions. You can use nucleic acid probes to look at protein-DNA interactions. You can use drug probes to look at protein-drug interactions. You can even do kinetics with this. So this functional microarray is very, very versatile, that is being used extensively for protein-protein interactions. So the purified proteins are spotted on the glass slide. In fact, the entire proteome can be spotted on the glass slide, and then you label the proteins present in a cell and then pour it over this glass slide and ask what kind of proteins is being bound to the immobilized protein. So the yeast proteome has been characterized by this method. Similarly, the calmodulin interacting proteins have been identified. So as I said, the protein-protein interactions, enzyme kinetics, protein-drug interactions, protein-DNA interactions, this is very, very versatile technique for analyzing the protein interacting partners of a protein. The second technique for studying protein interaction is the affinity chromatography with mass spectrometry. The affinity chromatography is basically where you are looking at a specific interaction between a ligand and its substrate. And so the recombinant protein is usually expressed as a fusion protein with an affinity tag either at the N terminus or C terminus. Many different kinds of tags have been identified: His tag, GST tag, FLAG tag, MIG tag. So you have a wide variety of affinity tags that are available. The cells are lysed under non-denaturing conditions so that you don't denature the protein, and often also you don't break the interactions between the protein. And the tagged proteins are purified using affinity chromatography and then further analyzed using either SDS page or mass spectrometry. So the affinity chromatography you can do two ways. One is where you have overexpressed the recombinant protein in E. coli cell, and then say it is tagged with GST. So you bind it to a glutathione column, then you lyse the cells, and your cell of interest, say you're looking at interacting partners in yeast, and this is an yeast protein. So you have you lyse the yeast cell, prepare the lysate, and pour that lysate over this bound protein. And then you wash away all the non-specific interactions and then analyze it on a SDS page followed by mass spectrometry. You always need to have a control column where you have not bound your GST tagged protein, but just the glutathione column itself, and you pour the extract over it. This will tell you eliminate all those non-specific interactions. So this is a very critical control. You can also do the other way, where you express the recombinant protein in yeast itself, and then you denature the yeast cell and then you pull it down. So you can do it either way. You can use one tag or you can use multiple tags. That choice is up to you. The multiple tags is preferred because it increases the chances of just obtaining the specific interactions, and it eliminates many of the non-specific interactions. But there is a problem with that. In the case is that if you do this multiple tags, then if you have transient interactions, that might get lost, because in case of multiple tags, what you have to do is you first purify with the first affinity tag, then remove it, and then do another round of purification with the second affinity tag. So if it is a very transient interaction, just for few minutes, or it comes off very fast, then what happens with many repeated steps of purification, that interaction can be lost. But the major advantage of this method is the proteins don't need to be labeled. Remember, in functional microarray, you need to label the protein. Here, you don't need to label the protein. And if you're expressing the protein in the same cell where you're studying, say yeast protein is being expressed in yeast, then the post-translational modifications will be retained. So that interaction will, and if post-translational interactions are necessary, then that interaction will be preserved. And finally, the dynamic nature of the interaction can be probed. But if you're going to use that where it is going to be expressed in the same cell, so yeast protein is being expressed in the yeast protein, care must be taken that you don't overexpress the protein. Because when we overexpress a protein, the interactions can be lost, alternate interactions can be formed, and so interactions that normally will not exist can start existing. So therefore, you might get a false positive result, and therefore you have to be very careful how we overexpress the protein. And so this is the major disadvantage, and you have to be very careful about it. So to summarize what we have learned in this module, we know that now that cellular processes, almost all of them, whether we talk about transcription, replication, repair, recombination, or metabolic processes, catabolism, anabolism, everything is mediated by protein-protein interactions. There are many factors that play a critical role in these interactions, and these interactions are characterized by certain structural characteristics. These protein-protein interactions evolved either to stabilize the protein or to optimize the efficiency of the function. Many techniques have been developed to delineate, to understand these protein-protein interactions. Of these, protein microarrays and affinity chromatography can be used to study the interaction at the genome-wide level to identify all the interacting partners of a given protein.